Everyone knows the five senses, but do you know that we have a sixth sense? It's called proprioception, and it is your sense of self. Proprioception coordinates and regulates posture, movement, and balance. And touch contribute to proprioception, but so does another lesser-known system, the vestibular system. The vestibular system is located in your inner ear, next to your hearing. There are 3 semicircular canals which each contain tissue called crista ampullaris. They regulate rotational movement of the head. There are also the utricle and saccule. They detect gravity, vertical motion, and linear movement. This system is what keeps you balanced during movement. They contain specialized cells, hair cells, and convert mechanical signals, head movement, to a chemical and electrical signal, which is carried to the cerebellum by neurons. Hair cells are one of the only types of cells in mammals, which include humans, that do not regenerate. As we age, the cells in our body are continuously being renewed, but not hair cells. This is evident in the elderly populations who have a higher probability of falling. Did you know 30% of people ages 65 or more fall each year? However, old age is not the only factor that can affect the vestibular system. Certain medications, called aminoglycosides, have the possibility to harm hair cells. While taking these medications, some patients may start to experience vertigo or the sensation of spinning. Unfortunately, we do not know what is occurring to the hair cells during this time, or why these adverse effects sometimes persist after ceasing treatment. This is where I come in. At the University of Barcelona, my doctoral research consists of understanding what is happening to hair cells and their synaptic connections with their corresponding neurons during these adverse effects. By using a chronic intoxication mouse model, I can chronologically observe hair cells while they are being altered, similar to prolonged use of these medications. Using biochemical methods, I can tag and observe specific proteins in hair cells and neurons to quantify their populations, locations, movement, and size. Using molecular biological methods yields gene expression data, and finally, we can observe ultrastructural data, which visualizes the physical structure of cells, synapses, and neurons. All of these data contributes in helping to construct a more detailed picture of damaged and healthy hair cells and neurons, and the roles of the proteins that are being affected by these medications. It also gives clues to the mechanisms being altered and possible signaling routes that may be intercepted in order to stop or protect against adverse effects in damaged hair cells and/or neurons. This research will also give insight to the general mechanism of hair cell death, and this has implications in aiding future research in hair cell preservation or even regeneration.